All right, everyone. Hello. Happy Wednesday. Welcome to FAU Harbor Branch Oceanographic Institute and our John and Barbara Ferreira Ocean Science Lecture Series. My name is Dr. Gabby McHenry, and I'm the Director of Education and Outreach here, as well as your host. So very warm welcome to everyone today. As always, I always like to begin our lectures by extending a very special thank you to those who support our programs, and especially those who have made a gift to support our Ocean Science Lecture Series, which many of you have this season. We are truly thankful for your contributions. As you know, there are many great ways that you can get involved and make an impact on the Institute, from volunteering your time to sharing what you learn with your family and friends, participating in our other programs, and of course making a donation to support our research or education programs. And I'd be more than happy to chat with you after the lecture if you'd like to learn more. And of course, I always want to thank all of the staff and our speakers who help with this series, as it wouldn't be possible without them. A quick reminder that you're all familiar with by now, all of the presentations are also available virtually, so hello and welcome to those tuning in on our live stream. And they're recorded, so if you missed any of our presentations throughout the series, or you had one that you really loved and want to watch again, maybe share it with a friend, you can go back and find it on our website. And for everyone here in person, don't forget after each lecture we host a meet and greet reception with our speaker in the gallery. We'll also have some cookies. And we invite you to stay, mingle, visit our pop-up gift shop, and learn more about our programs and our research. So we've had a fantastic season and we're in the final stretch. Our last lecture will be held next Wednesday on March 26th, and that is titled Blue Energy in a Blue Economy by Gabriel Alcenis. And he'll share some really great insights into marine renewable energy and the latest innovations to generate power from the Gulf Stream just off our coast. So you won't want to miss that. And today's lecture is titled Ink-redible Investigations, What Sea Slugs, Cuttlefish, Whales, and Sharks All Have in Common by Dr. Lauren Simonitis, who is a research and biological imaging specialist for Florida Atlantic University's on-site lab school, A.D. Henderson University School, and FAU High School. Dr. Simonitis received a dual bachelor's in marine science and biology from the University of Miami and a PhD in marine biology from Texas A&M University. She was formerly a National Science Foundation postdoctoral research fellow in biology at both Florida Atlantic University and the University of Washington. As a sensory biologist, Dr. Simonitis is interested in how animals use their sensory systems to understand their environment and navigate predator-prey relationships. From the prey perspective, she focuses on how animals use ink as an anti-predator defense. She also studies the sensory systems of a common ocean predator, sharks, employing a suite of biovisualization techniques. And in partnership with her colleagues, Dr. Simonitis manages the current FAU High School Owls Imaging Lab, which serves as a biological imaging center to facilitate research, collaboration, education, and mentorship. And she's also overseeing the planning and management of the soon-to-be-open Marcus Research and Innovation Center. Additionally, Dr. Simonitis oversees the school's curriculum integration, infusing hands-on, cutting-edge laboratory experiences into the science curriculum of A.D. Henderson and FAU High School students. So I met Lauren last year. She gave a wonderful presentation at the Florida Marine Science Educators Association conference, and I immediately said, we need to bring you back to Harbor Branch to present at our Ocean Science Lecture Series. So I'm so excited for you to hear about her work, and please join me in welcoming her.
Awesome. Thanks, everybody, so much for having me today. I'm so jazzed to talk to you guys about my two favorite things, which are shark noses and ink. And you might be thinking that there's no way that those two combine, but I will convince you otherwise as we go through. Thank you so much, Gabby, for that introduction. And I'm so excited to get started with you guys. So As a scientist, I employ a couple different techniques. So we've talked a lot about imaging in my background. I use a lot of that, and that is where I look at morphology. So that's how things look or how things are structured. I also look at physiology, so how that animal's nervous system or their body systems are making it function. So I look at what it looks like, then how it works, and then how those two things come together to give us behavioral outputs. So what does the behavior look like when that morphology has physiology and then impacts behavior? So I'm going to kind of go through all three of these when I talk about my work today. But really, my whole interest here started with inking. So what you see here, this is Simone. She was a pygmy sperm whale that was in managed care at Moat Marine Labs, so not too far from us. And what you can see here is she is inking, and she is inking directly out of her anus. So a lot of animals will excrete waste when they're scared or when they're overstimulated. So a dog gets really excited, it can pee a little bit, or you pick up a frog, it doesn't want to be picked up, and it might excrete some substances at you. That's pretty common, but Simone here, she has infused a little bit of extra pizzazz into her poop, and that is ink. And here you can see it from above. You can see it's pretty effective. So it covers a lot of this tank. You'll see her swimming into it and hiding in it. And she basically disappears into this ink if you're looking at her from above. So what even is ink, right? So there's a couple different groups that use it. Today we're going to focus on three. So the first are cephalopods. They're probably our most famous of inkers. We have squids, octopus, cuttlefish. We also have sea hares, so we might see them around us here in South Florida, and we're going to talk about the different species that use it as well. We also have pygmy and dwarf sperm whales. So to kind of give you a little bit more of an explanation on either of these, displays. All right, well, if this video was playing, oh, there it goes. Okay, so here you can see how cephalopods use their ink. And cephalopods inking is really cool because they have an extra little suite of special skills where they can change their coloring in order to match that ink. So inking is a very big visual display. So what I want you guys to see here is how right before the cuttlefish inks, it's going to turn this dark black color. Then it's going to go through that ink cloud, keeping that dark black color. That helps with that camouflage. And then it changes more into that transparent sand-like color when it swims out of that ink cloud. It's really easy to talk about my work if somebody has seen Finding Nemo, and they always quote that little pearl guy to me. So their ink is based in melanin, so it's the same thing that gives our hair color, our eyes color, our skin color, and it's mixed with mucus. So what you're going to hear when we talk about ink is it's kind of this combination, this chemical cocktail of two substances. So the first is the pigment, which is what gives it color, and the second is what makes it goopy, and these guys are mucus. So they have an inking gland and a mucus gland, and then they use both of them together at the same time, and they can actually modulate how much mucus and how much ink goes into a display. So over here, this is from a paper where they looked at different inking displays in these squid and matched it to their behaviors. So if they put just a little bit of mucus, they can kind of create these more cloud-like appearances. If they put a lot of mucus, they can create these ropes. They can also create something called a pseudomorph, which we'll talk about in a little bit, but it is a modular response. So it's not just the same thing happening every time. They can kind of finesse their ink responses. Next are sea hares. So this one here is Aplysia californica. So you'll see them off the California coast. And you can again see them inking over here, and you can see that they have these ink clouds, but they also have these rope-like structures. And these guys have been with me for a very long time. My first ever research experience was in the Aplysia lab at the University of Miami. I was studying their nervous system because they're great animals to study neurobiology. They have a beautiful nervous system, a simple nervous system. Eric Kandel won the Nobel Prize studying them. And it's been a really long journey with them, and I'm very happy to still have them. Before, I really hated when they inked because I couldn't see the neurons, and now I am milking them for their ink. So they have a red algal-based ink. So if you stop feeding them algae, they will no longer produce that red color. What they do is they take that pigment in the algae, they modify it within their own bodies, and then they put it into the ink to give it that red color. They'll still produce the rest of the chemicals in the ink. It'll just be clear. But their mucus, sticky part, is opaline. So it is from a different gland, and when it comes in contact with water, it polymerizes, gets real nice and gloopy, and you can kind of see these two responses here. So this is a response that is heavily ink, very little mucus, very little opaline. So you can see it's not as bubbly, and it's really dark purpley pink. And then over here, we have our lighter opaline heavy response that is very bubbly and not so dark in color. My amazing intern, Anna, she's now getting her PhD in Canada. She did a study where she looked at how these little slugs can actually learn what is the best way to combine their inking responses to avoid a predator. So if we bothered them every day, and one day they tried a lot of ink, a little opaline, and the next day they would try a lot of opaline, a little ink, and then we stopped bothering them, every time we would go to bother them, they'd give us the same reaction that stopped us from bothering them. So these little slugs are really impressive, even with their really simple nervous system. So here's kind of the stars of the show, the pygmy and dwarf sperm whales. I'm going to refer to them as the Kogias for the rest of this talk. And then in my last talk, the last slide, I'm going to talk about the sea slugs as aplesia. It's a little easier to say. So these guys are really neat. A lot of times I used to work on stranding teams, and we would get calls for them, and people would call them in as sharks because they have these false gill slits, and they kind of have these blunt heads, these subterminal mouths, which you see a lot on sharks. But like I mentioned, they release a mix of ink and feces. So here's me getting a jar of feces in the mail, one of probably very few people that gets super excited when I get a jar of poop on my door, but it's a great day for a biologist. So for my PhD, I really focused on a representative from each of these three groups. And I wanted to compare their ink to see how has this evolved over time. So how does it work? It is an anti-predatory defense, which means it stops predation. But in order to understand what an anti-predatory defense is made up of, we need to think about the 4 steps of a predation event. So first we have the initial encounter, and I'm going to kind of describe this from the perspective of the predator and think about what kind of senses are being used. So this initial encounter is using long-range senses. So if the water is clear, that can be vision. It can also be smell and hearing in the aquatic environment. Then we move into the attack. So this is when the predator is approaching the prey. And again, we're moving from those long-range senses into those short-range senses as we get closer. This is where we get the capture. So this is when the predator has the prey within its control. It is now using its super close range senses, like its mechanosensory or its touch, as well as its gustatory or its taste. And then finally, we have ingestion. So that is the prey is within the predator's mouth. It's moving through its digestive system. But there are still some defenses that the prey can have at this point. So if we flip this around and think about it from the prey's perspective, we have general versus acute threats. So in the acute threats, that is the latter half here. That's where we can have defenses such as helmets or helmet teeth, which are these little spikes that come off of helmets on some animals. We can have shells. We can have hard exoskeletons. We can also taste really bad, have bad tasting skin, slimy skin, anything like that. But it's a lot easier to defend yourself before you're in a shark's mouth. So we also like to think about these general threats and how we can stop them there. So these are where we want to attack those long-range senses. We want to attack vision in a clear environment, but we mostly want to attack this longest range, which is smell. So ink is great because it works on 3 sensory systems. 1, visually, you guys saw Simone hiding in her ink. It is a very dramatic visual response. They're often, it's like a big Houdini cloud. It also has some chemicals in it that smell really distasteful and aversive. And also, like I've mentioned a couple times, there's a goopy part of ink, and that is where the physical discomfort comes from. So it can get all on your chemosensory appendages, and then it really attaches that bad smelling stuff to your nose, to your smelling appendages. It is a really great weapon. So ink is used a couple different ways, and I'm going to kind of go through 3 of them. So the first is phagomimicry. So this is when we have a squid kind of swimming around. All of a sudden, it's going to see a predator. It's going to use its long-range senses to sense a predator. Like I mentioned, one of the great things about inking in squids is that they can actually use their chromatophores in order to change visually to match the visual signal of the ink. So they'll change to this dark black coloration, produce what we call a pseudomorph. So this is a lot of mucus and a lot of ink, and it creates a little decoy squid, a little ink cloud. Then it'll change into a transparent coloration and jet away. And from the shark's perspective, it saw a dark squid. There's still a dark squiddy-like shape. But then the shark is going to go take a huge bite of that ink and realize that it is not a squid. So this is where the phago part of this comes in. Phago means smell. So it is also thought that maybe the ink has some nice smelling stuff to entice that shark in order to bring him in and say, hey, this is a nice smelling squid. Another thing that ink does is it acts as an alarm cue. So this is a way that communities can kind of defend themselves. It basically helps sound off an alarm that says that there's a predator in view, a predator is coming, let's all get out of the way. It's a version of community care. So these squids and these cephalopods and these very, very sessile sea slugs can actually swim really well when you entice them right. So one of the best ways to get sea slugs to swim is if you drop a little bit of ink on them, they will actually start flapping their parapodia and swimming because they're saying, well, if Frank over there is inking, that means that there's a problem, so I should probably get out of here as well. The last thing that ink can be used for is defensive chemicals. So the best example of this is the skunk. Smells bad, is not fun. You no longer want to eat the skunk when you are covered in the skunk smell. So really effective stuff. So those are kind of the 3 ways that we think that ink works. But what I found really interesting when I was studying this was I think that ink is a really awesome example of what we call convergent evolution. And convergent evolution is whenever there is a problem that unrelated organisms solve by the same route without having any evolutionary passing down of that trait. So between ink, there's no giant old ancestor of slugs and cephalopods and whales that all figured out this inking response and passed it down. They've all developed the same solution to the same problem independently. A great example of convergent evolution is echolocation. So both bats and dolphins have overcome a lack of visual cue or lack of being able to see by developing the same response of sending sound and measuring how it comes back. So once again, we're going to look at these 3 individuals from Aplysia californica, from Kogia, and from Cuttlefish in order to see how has this ink all convergently evolved. So have these guys all kind of evolved the same response to this predator, and are they all effective? So do these 3 inks that are unrelated have a similar anti-predator effect? And I picked the very illustrious and terrifying bonnethead shark in order to fill in that gap. And if you've ever seen a bonnethead shark, you know that they're about 3 feet long on a good day if they're standing up extra straight. So they are not exactly these hyper predators that are eating whales. But one of the really great things that some of my collaborators have found is that no matter what shark you test, if you stick a little electrode in their nose, they all have the same smelling abilities. So if you test a great white shark and you test a bonnethead and you see how sensitive they are to certain smells, despite their noses looking pretty different, they have the same smelling abilities. So we can use bonnetheads, which are small. They have giant noses hidden here in their cephalofoil, and we'll talk about those in a minute. And we can apply what we learn on them to some sharks that are actually eating whales. The great thing, too, about bonnetheads is that they hunt squid, so inking is something that they would encounter in their natural environment. It's not something that's completely novel and something that they would never encounter. So now I have kind of the other end of the spectrum, and I know what I'm going to use as my thermometer. So now I study a lot of sharks, and a lot of people kind of assume that I'm a shark person. I just needed a good thermometer. That's all I needed, and I kind of stumbled my way into shark science. So Now that I have the other side, I'm ready to ask this question. Do these three unrelated inks have the same response? And I'm going to use that behavior part of my toolkit. So I have this free-swimming shark right here, and this is an unmotivated shark. It is not being fed. I just wanted to see the natural response of a free-swimming shark. So in order to do this, I took ink from Aplysia, Cuttlefish, and Kogia. And unfortunately, when you get ink, it does not come with a known concentration of how much ink is in every liter. So we had to figure out, how do I standardize by volume and by concentration between a slug that gives me 1.5 milliliters to a Kogia that can give me up to 5 liters at a time. So we standardized the volume, so every time we were putting 5 milliliters in. We did that because when I put that in, that's about the size of an ink cloud that a shark will swim through and get its nose all in. And then we calculated how much dissolved organic carbon is in each of these 3 inks, and we standardized it so that every single time we put the thing in the water, the same amount of carbon was going in. We also wanted to make sure that there wasn't something affecting the shark besides just the ink itself. So we had a couple controls. The first is seawater. We put seawater in the water just to make sure that getting something shot into the water wasn't bothering them. We used food coloring to account for the visual signal. And then we had food odor as well. So food odor, one, it gave us a positive response. So we could see the difference between what we thought was going to be a negative response and a positive response. It also helped us make sure the shark's nose was still smelling. And what I mean by that is, I'm sure that many of you guys have experienced this. You walk into somewhere, a bakery, it smells like muffins, it smells really good. But then the more time you spend in there, you get acclimated to it. So your nose kind of desensitizes. Every single time that we expose the sharks to this smell, their nose is getting less and less and less sensitive. So as long as they were still responding to food odor, we knew that their nose was still testable. The minute they stopped responding or the minute that they started avoiding food odor, we knew that all of our behavioral trials since the last appropriate response were not appropriate. Sharks are not very emotionally available. They can't really tell you how they're feeling. So we need to invent ways as scientists to make sure that the data that we're getting is the actual behavior that we're interested in. We had a camera filming overhead. We had 5 sharks. And we looked at a couple different locomotory kinematic variables. So that is a big word that means how the shark is moving through space. And I'll kind of go through what an experiment looks like and what one of those variables that we looked at today. But first, you can see here that in this picture, I am painting little dots on my shark. So if you've ever seen how video games are made or CGI in films are made, they wear those ping pong ball suits that look real funny. But I did the same thing to my sharks, so I painted these little white dots on them, and that allowed me to digitize these points in the shark. So as the shark moved, instead of looking at all the parts of the shark, I got a little stick figure shark, and I could see how fast it was swimming, what angles its body was making, the way that it deviated from where it was swimming. All these locomotory kinematic metrics were taken off of this little stick figure shark. Okay, so this is kind of how an experiment went. So this is if you are my camera, you are sitting overhead of my tank. Here is the shark. Here are the injection sites. So these are little blue discs that sat at the bottom of my tank. Then we had kind of these little straws that came up and sent ink into the water column. And then these tubes ran to where my amazing interns were crouching down so that the shark couldn't see them. And I was on a platform overhead. So whenever the shark was about 1 body length away from the injection site, we started to release the ink. So much so that when the shark swam through that ink cloud, it had a nice olfactory flushing. So I mentioned that we looked at a lot of the different kinematic metrics, but today we're really only going to focus on this one. So this is angle of deviation. So this is going to tell us how much did the shark change its swimming path in response to the stimuli in the water. So over here in the first panel, you can see we have A before the injection and B right after the injection. So we drew a line right down the middle. And this was the shark's initial bearing. So here is where you can see the initial little bubbling of seawater. And then you see there's not really a response. So it's about a 20-degree variation because sharks do swim sinusoidally. They swim back and forth about 20 degrees either way. So that's not really a response. Over here, we see kind of the opposite. So here's that initial bubbling, and then you can see over here the resulting big ink cloud. And you see the shark made an over 180-degree response away the opposite way. If the shark turned towards the stimuli, it was recorded as a positive. If the shark turned away from the stimuli, it was recorded as a negative. And we had some pretty striking results. So to kind of orient you here, this is the maximum angle of deviation. So this is how much that shark turned away or towards the stimuli. Here we have A, C, and K, Aplysia, Cuttlefish, and Kogia. And they're all colored purple for their ink, and they were all overwhelmingly negative responses. They were significantly different from our two controls of food coloring and seawater that had very little deviation, and then we had an overwhelmingly positive response to food odor, which was great. It was really nice to see these two behaviors be so different visually and then also statistically. So overall, we saw that ink elicited an avoidance response, and you can kind of see that on this video right here. This was great, and it was different than food odor. So if you think back to phagomimicry, where I talked about how one of the theories about how ink works is that it also kind of entices the shark in chemically as well as visually, that was not what we saw here. You can see here this video where she is responding to food odor. So she swims into that nice cloud and turns back around. And this is great because when we think about Simone, she goes back in and she hides in her ink. If this was a phagomimetic response, if this was something where we're trying to entice the shark back in, this would not be the best strategy in order to hide in what is enticing. So it makes sense for the animals that we're looking at. So at this point, we kind of had a good response over how these inks are responding, are making the shark respond. But this was just behavioral, and what we wanted to do was get further into the physiology. So what was going on inside their nose that was making this reaction happen? So are they actually smelling the ink? And this is where physiology comes in. So this is kind of our Frankenstein shark situation that happens over here. So we have our shark lying back, and we have electrodes that go into their nose. So whenever a chemical binds to an olfactory receptor neuron, it sends an electrical signal that says, hey, I'm smelling something, and that signal travels through the nerve up to the brain. What happens over here is if we put an electrode right next to it, it connects to our computer, and it gives us a nice depolarization that shows us that the nose is responding to chemicals. And we can measure how heavily that response is happening in order to test how sensitive the shark's nose is to those chemicals. We can also see how much these chemicals affect sharks, so it kind of works both ways. So I was supposed to start this experiment in March 1st of 2020 as the last part of my PhD. And then I never went back to the lab, so that didn't happen. But I wrote it up as my postdoc, and I came down here to Florida, and I did this work in the Florida Keys. So this is work that we're currently analyzing and hoping to publish soon. But this is our shark right here, and then this is me finally getting my first response 3 years after I started these experiments. And this is going to be a little video showing you how this response works. So you're going to see us testing to make sure that the water is going into the nose. So you'll see a little blue dye go into the shark's nostril and then come right back out. And you can see that there's a tube in its mouth so that it can continue to breathe throughout this experiment. Okay, so that work is ongoing, but we have a pretty good handle, and I can just tell you from looking at that data that it matches the behavioral experiments really, really well. They have huge responses to the ink, and the level of their response matches the level of their behavioral response, which is really fun. So I'm really excited to get that publication out soon. So I also did some work looking at the chemistry of these three inks and looking at what chemicals have been chosen to be part of this chemical cocktail every single time. So I've kind of looked at the makeup of the ink, I've looked at the interaction of the ink, but my next part was going to be looking at the receptor of this ink, so looking into a shark's nose. When I started this work, I was kind of under the impression that we knew a lot about shark noses because they're so famous for it, But actually, the literature is really, really sparse. We do not really know that much. It was a really hot topic item right after World War II because of the interest in shark deterrence, but then it really died off. So there really hasn't been too much work done recently, and my colleagues and I are hoping to revive it. So to kind of give you a little tour into a shark's nose, this is the bonnet head once again. They are my favorite, if you could not tell. So what you can see here is these beautiful pores. Those are their ampullae of Lorenzini. That is what is responsible for the electrical signals. But then, if I can draw your eye away from them, here are their nostrils. And I say nostrils because they actually have 2 nostrils on either side of their head. So this right here is their in-current nostril. This is where water flows in. And then here is their excurrent nostril. So water, as they swim through the water, water flows in through the in-current nostril. It follows this little groove. It goes into their olfactory chamber, which is inside their head, turns back around, and then is shot out this excurrent nostril. So it's a nice flow-through system. It is also not connected to their mouths. So actually that nose-to-mouth connection, we don't see that until animals move on to land. So until amphibians, we do not have a nose-mouth connection. They cannot sniff. They cannot blow their nose. They move water through their nose passively the same way that they breathe. So if we are going to flip that shark over and take a look inside, this is what we see. So like was mentioned, I do a lot of biological imaging. And this is a contrast-enhanced CT scan. So normally in CT, we only see hard parts. Excuse me. So here we can see all the soft parts because I soak it in this radio-opaque liquid. This part right here is that beautiful smelling organ. So this is a CT scan that we are moving through the shark's head. And you can see the in-current nostril right here that leads to the in-current chamber. So this is where water is going to flow in and travel to the inside. And then here, it's going to be coming out the outside, the excurrent chamber and the excurrent nostril. So this excurrent chamber terminates in this excurrent nostril right here. What's another great thing that we can do with CT scans is we can actually pull out the part that we're interested in and make it a 3D structure. So this is what that rosette looks like. So this smelling structure is called the olfactory rosette, and that's because it kind of looks like a flower to some people. All of these individual petals are where the smelling happens. I think they kind of look like dishes stacked in a dishwasher. We can also turn it to the side and look at one of these dishes. So this is if you took one of those out, looked at it, all of this part right here, these are called the lamella, and they're a paired structure. Inside of all those lamella is where those olfactory receptor neurons are. Those are what I was talking about before. So water is going to flow across the structure and through the structure. It's going to bind to the olfactory receptor neurons and send off those electrical signals that says, hey, I'm smelling something. And then we can look at the different parts of this to tell where the smelling is happening. And we can do that through a variety of techniques. Today, I'm going to talk to you mostly about the one on the right, but I love histology, so I was going to talk to you about it, which is on the left as well. So over here is what is a histological slice. So basically, we get soft tissue, we put it in a hard plastic block or a hard paraffin block, like paraffin wax, and then you can slice it on essentially a very fancy Publix deli meat slicer, but it cuts down to 5 microns, which is thinner than your fingernail. So we can actually see cell layer by cell layer what makes up this structure. So that can tell us where the sensory tissue is. We can also see it in scanning electron microscopy, which is on the right. This is giving us a really high definition vision of what is on the surface of the structure. So we can also divide it into area that is involved in sensation, so sensory area, it's called SA on this slide, and non-sensory area, non-sensory area. So to get a little closer in the scanning electron microscopes, you can see the non-sensory epithelium over here and the sensory epithelium over here. This over here is the olfactory nerve layer. So all of the olfactory receptor neurons out here in the sensory epithelium send their axons through the olfactory nerve layer, which travel up to the brain. If we get a little closer, you're going to really be able to see the difference. So here is the sensory epithelium and the non-sensory epithelium. Sensory epithelium, it has all these little ciliated cells. They kind of look like spaghetti. And spaghetti is a great term because the olfactory receptor neurons sit, if you were to like part the spaghetti, you would see little meatballs. They honestly do look like little meatballs. And that is where those chemicals bind and send that signal to the brain. The sensory epithelium, that ciliated part, they move mucus along. So a lot of my work is actually just looking at shark boogers. A recent study came out, and there was actually a follow-up study to this where it actually informed how we make washing machines and how we make high-performance washing machines because shark noses are so good at filtering water efficiently. But what's really cool about this study is they looked at a very similar to a bonnet head, a small eye hammerhead. And they found that in the middle of their organ, they have this really long tube-like organ. They have high rates of water flow, so you can see the red and the green, these higher areas of high water flow. But then they had really low water flow in the ends of them. So at the medial part, so the part closest to the middle of the head, and the external part, so the part closest to the outside of the head. These are the areas that water flows less, and here water flows more. And then when we look at those sensory area versus non-sensory area, we found that it matches where the water flows differently. So areas of high flow in the center, they have more sensory area. And areas with lower flow on the outsides, they had lower sensory area, which makes sense. If you have a lot of water coming by quickly with a lot of chemicals, you need a lot of places to catch those chemicals. But then when you go on the sides where water is moving slower, you don't really want to put as much investment into catching those chemicals out there. But a hammerhead is a really unique animal, even amongst sharks. There are not that many sharks that have these really elongated tube-like noses. So I started going more into looking at different types of noses and different types of sharks. So has anybody here ever dissected a shark before? Yeah. Some of you guys have. And it's really popular now in schools. So a lot of schools and a lot of summer camps will dissect spiny dogfish sharks. And these are these guys right here. This is the Pacific version. So he has a nice little pointy head, and he has this really cool spiracle on top, which is what allows them to pull in water and irrigate their mouth, even when they're not swimming. It can also help irrigate their nose. So if we look at it, it has the same type of distribution. So it has these dishes in a dishwasher. But when we look at it in 3D, it creates kind of more of this ball-like effect. So I like to think about this as like the tube to ball ratio. We have very tubey-like noses and very ball-like noses, and then we have a lot of noses in between. And then we can get even wackier. So this guy over here is a spotted ratfish, and these are chimeras, and they really diverted pretty early in the evolutionary history from sharks. So they can be kind of representatives of what early sharks looked like. They're often referred to as living fossils. They also have two front-facing nostrils, which is interesting because it's kind of the opposite of what we see in most sharks. So we think this might have been the ancestral condition. So we started with two front-facing nostrils, we went to two side-facing nostrils, and we kind of worked our way back to the original design. Their noses are also very fun. So if we look inside, we can see something very different. So this is going to be a video. You can see it's two nostrils facing forward, and then we're going to slice in. And we have these lovely little daisies. So instead of dishes stacked in a dishwasher, they're dishes arranged radially, which I think is how my boyfriend stacks the dishwasher many times. Does not make any sense. So over here we can see what these two look like inside of the nose. So we can see this ball and we can see this daisy. And then we can pull them out in 3D and look at them again. So if we look across, we see those dishes stacked in the dishwasher in a little ball formation versus dishes in this radial formation. And now what we're looking at is we're trying to fill in all the diversity of noses. And we're trying to understand, does that same pattern that we see in the bonnet head, where there's a difference in water flow and a difference in sensitivity, does that carry over to all of the different shapes and sizes of noses that we see across the sharks and their distant relatives? We can also now in 3D pull out those individual dishes, those individual lamella, and look at them both in 3D and then combine that with the high definition sensory tissue that we see in the scanning electron microscope. So here are two of those Pacific spiny dogfish, binded in the middle. And then over here, we see one from the ratfish. So where our research is going now is we're looking at the big diversity of shark noses. We're trying to see that morphology, so looking at the function, the form, and then we're going to get into the function. So we're trying to do more computational fluid dynamics and more fluid studies to see how these different noses have water flow through them. So we really only know about this tube. We don't have any more information really on any of the other morphologies. We have some guesses, but all of the work has been done on this one really long nose. So we're trying to get into this field. It's also really interesting from a device perspective because shark noses, they are really highly sensitive. They can smell really, really well. And as far as we know, they do not clog. So we're hoping that it can help inspire some devices that are really great for filtering liquid and detecting really small chemicals without clogging. We're going to continue our physiology experiments, not just testing different inks, which I would love to keep doing, but also varying the water. So every physiology experiment we do is based on a hammerhead because that is the only shark that we've really calculated their flow rate for. So we're thinking that maybe if we change the flow rate that we're testing their physiology to actually match what the shark is doing, we might get some better responses. And then we're going to continue our behavioral work. So looking at how different smelling cues affect how that shark swims. So we really want to flesh out this entire picture. But really, it just all comes back to these three parts, morphology, physiology, and behavior, and it all comes back to ink. Because what I'm really interested in is I would love to understand how water flows through a nose. That's cool. But what I really, really want to know is how does ink work? Because it is goopy, it is thick, melanin is a huge molecule, it clogs things up, it clogs up filters, it is a mess. And even just from watching my sharks after I introduced ink into the water, like I said, they can't really blow their noses. So what they do is they swim around really fast in the tank trying to flush out their noses. So I really would love to understand how water works just so I can get back into the ink, back into the slugs. I also just wanted to take a moment to talk about a nonprofit that I work for. So this is Minorities in Shark Sciences. We are based in Florida, but we have members from all the continents except for Antarctica, even though one of our members is there right now doing research. This organization is super important because there was actually a study that looked at all of the presenters on Shark Week, and there are more people on Shark Week that are experts named Mike than there are women. So we are trying to change that because we exist and we do work and we are fun and awesome and great at speaking and great at talking on TV as well. So we offer these experiences for our members to do research completely free. We pay for everything, including their visas, especially if they're from the global south, because that's another huge problem in shark sciences is there's a lot of programs where you pay a lot of money to go and maybe touch a shark once, but mostly you're washing glassware. And these are really cost prohibitive to people that can't get those experiences. So we offer experiences to our members completely free. We pay for conference travel. We sponsor research. We write grants. We also have a lot of free educational resources for the public. We have an entire educational program called Gill Guardians. And I'm super grateful to them because they have made me feel really at home in shark science. I used to not call myself a shark scientist because I did not see any shark scientists that looked like me or acted like me. So they have made me call myself a shark scientist again, and I would really love if you guys check this out. You guys can applaud from this. I also wanted to just quickly talk about, Gabby mentioned the FAU High School Research Program. I run an imaging lab. You can check us out on Instagram, but we are a training facility, which means that we train our high school students, but we also train other researchers as well as our elementary students. So these are our K-5 students learning how to behave in a lab, and they even come in and use our scanning electron microscope. So these are two of my fourth graders that found a rock outside that they thought was really cool. They were doing a rock lab, so we stuck it in the SEM, and they actually got to touch and drive a scanning electron microscope, something that I did not do until the 6th year of my PhD. So I'm very happy that I got a job before these guys graduate because they are definitely more equipped than I was. And with that, thank you guys so much. I will take any questions. Yes. Yeah, the main one is those filtering applications because we really don't understand how this system even works. A lot of times I bring 3D models that I've printed from sharks to engineers, and they say, this does not work the way that you think it does. And I'm like, well, they've been doing it for like a really long time, so it is working and it is effective. I think that's one of the biggest ones. From a conservation standpoint, we really need to understand how chemical communication works now because, as I'm sure many of you know from listening to these types of series, our ocean chemistry is changing. And from the limited work that we've done, we've seen that in future ocean conditions, when oceans acidify, it can actually affect how sharks and other animals respond to chemical cues. They start avoiding prey response, and they start avoiding prey cues, and they start going towards predator cues. So it's actually messing up their ability to smell and respond to those smells. So it's really important to understand what's happening now at baseline so that we can predict what will happen in our future ocean conditions. Thanks. Yes? Yeah, so some of those that you saw were actually interspecies displays. So they can talk to other species, or they can also talk within their own species through their ink displays. The other thing about having really heavy mucus is we see that a lot in spiny lobsters. Spiny lobsters are mostly who have been tested as victims of ink. It covers their appendages, and then they spend a lot of time doing their little lobster cleaning instead of attacking the inking animal. So it will bind and cover all of their appendages, make it really nasty, but it also helps them make all those different displays for communication within their own species and between species. Yes?
You mentioned that after World War II there was an interest for shark research.
Mm-hmm.
Is that still ongoing?
Well, I have submitted some grants to the National Department of Defense, and they keep not being super interested in it right now, but I will get them. Don't worry. I will convince them that this is important. A lot of it has switched to more sonar-based. It's also switched to electromagnetic defenses. So there's different avenues that people are investigating, less so on chemical, because it's a lot easier to have a really high-powered magnet now than to have a constant stream of chemicals. But I still think there are some cases where that might work a little better for you. Yeah.
Sharks get close to a prey and they shut their eyes, cover their
Mm-hmm.
eyes with membranes. Does that have any?
So these guys actually don't have those membranes. And then there are some sharks that don't have membranes, and instead they'll roll it into the back of their eye. They roll their eyes into the back of their head like we see in great whites. Bonnet heads don't do either, which is one of the reasons why I chose them as well. But through their nictitating membrane, we actually don't know how much they can see because they're somewhat transparent. But also, when sharks get really up close, we don't think that they're very visual animals. There's been some other work done by people who study shark olfaction, and they actually put little blindfolds on them to see how much vision plays in. And when they get into that close time, they rely less on vision. That is also the reason that we used food coloring to make sure that the color wasn't what was bothering them. Great question. Thank you. Yes, it did. The government's interest in shark repellents did indeed come out of the Indianapolis. Yeah, it was there's a lot of specials about it, very interesting specials. It was a operation that a lot of sailors got stranded, I think they were Navy or Marines, I can't remember right now, but they got, their ship sank, they were stuck afloat, and many of them were eaten by sharks, and they think they're oceanic white tips. Yeah. That is a great question. Have they ever come up with a real effective shark repellent? I think my ink is pretty effective, but also Dr. Kajura at FAU does a lot of shark repellent work, and he is working on strong magnets that use their magnetic, they interact with the ampullae of Lorenzini, their electrical systems. So that is currently what's being tested. There's a company called Shark Bands that they sell like little bracelets. Those are not super effective, but what we're testing in Dr. Kajura's lab are these big magnets. They're like kind of like shaped like zeppelins that you can hang on your fishing lines to keep sharks away, because they don't affect fish, because fish don't have ampullae of Lorenzini, teleosts bony fish, but they do affect sharks. So those are proving pretty effective. There's also some work, some of my colleagues in Australia are working with really strong electrical pulses to do the same type of thing. Still ongoing, but none that I would like put my name behind and support right now. Yes.
What's the internal organisms for these ink-producing creatures? What do they all have to make the ink to secrete it? Do they all have similar endocrine system or some sort of a gland?
Yeah.
Organ?
Great question. So in Aplysia, the sea slugs, they have an ink gland that produces it. They produce it within glands. Within that gland, they have little production sites for the defensive chemicals, and then they take their the food particles, the red algal pigments, and incorporate it to do the color. In cephalopods, so in squids and cuttlefish and octopuses, they use melanin, which they already produce in their body, and they have a melanin-producing ink gland, and they have a mucus gland, and then those together come out through the ink tube. But something that's interesting is that melanin is a really good binder for heavy metals and toxins. So it's thought that they started extruding melanin and getting rid of it as a form of detoxification to get rid of the heavy metals that they uptake in their body. Kogia, we're still learning, but some work that was done in my lab, we sectioned their digestive system, and we saw that they have these melanin-producing vacuoles. They're like little melanocytes in their colon and in their jejunum, which is part of their digestive system before the colon, where they have these little ink-producing sites that ink directly into the jejunum and the colon, and then whenever they release the waste, it's mixed together. I also get asked if they can do one or the other. We do not know. I do not think so, but just like from logical sense, it seems like it just goes into their colon, and then whenever they poop, ink comes out as well. Yes. Yeah, I mean, I can just say, so the question was, are the cutbacks going to affect my research? I can say that they're going to affect everyone's research. I was funded, you can see National Science Foundation right there. They are my main funder. They were the sole source of my income and my research in the past 2 years as a postdoc. So that program is on the chopping block and is being cut. And if this was a year ago, it is very likely that I would be unemployed right now. So yes, it is. And it's affecting anybody who gets funding from the National Science Foundation or the National Institute of Health. Thank you for that question.
All right. Well, everyone join me in thanking Lauren for her amazing presentation. Very interesting and excellent visuals, too.
Thanks.
Awesome. Well, we'll join everyone in the gallery for any additional questions and munch on some cookies and chat about noses and ink. Don't forget to join us next Wednesday for Blue Energy and a Blue Economy, and that'll be our final lecture of the series. So thank you all so much for coming.